Our biology is controlled by DNA, our genetic instructions. This DNA is like a huge instruction manual with 3 billion chemical bases that determine what we look like and how our bodies work. But just one mistake in this genetic code can stop our cells from working correctly. Until last decade, molecular biologists could only observe these tiny chemical errors, but they had limited tools to intervene and correct them. Then came along Harvard chemist and biologist David R. Liu. He developed game-changing molecular tools, operating at the scale of individual atoms, to correct or replace a huge variety of genetic mutations. In 2025, he received the $3 million Breakthrough Prize, the so-called Oscars of Science, for his contribution to life sciences. He is a Howard Hughes Medical Institute investigator who created base and prime editing techniques now being tested in dozens of clinical trials worldwide. But he is also one of world's top players in Street Fighter, and someone so advanced at the mathematics of blackjack that he is banned from MGM Grand Casino at Las Vegas for winning too much. One of the therapies he designed recently saved the life of a 13-year-old British teenager named Alyssa, who suffered from fatal leukemia. His colleagues call him one of the most extraordinary scientists they have ever met. At the age of 26, he was already a professor at Harvard. His work has led to the creation of multiple startups which are already helping patients worldwide, and he has earned more than 100 patents, an unprecedented number for someone his age. This is the story of why he is considered one of the biggest contenders for a Nobel Prize this decade, and perhaps the most groundbreaking biologist you have never heard of.
Every mentor who taught me that breakthroughs with impact begin with compassion.
Liu was born in Riverside, California in 1973. His parents had immigrated to the United States from Taiwan. His father worked as an aerospace engineer, dealing with the physics of flight, while his mother taught physics as a university professor. From the very beginning, he spent his early years intensely observing the natural world with boundless curiosity. He foraged among the bugs and weeds in his backyard. He mixed baking soda and vinegar and other simple reagents merely to watch the physical reaction unfold. He wanted to ask and answer his own questions about the mechanics of physical reality. He later recalled that he liked chemistry before he even knew what the word chemistry meant. He believes most kids are intrinsically interested in science. He would spend hours tracing the chemical pheromone trails left by ants marching across the dirt. By high school, he began winning national competitions. He entered the prestigious Westinghouse Science Talent Search in 1990. He built a complex model simulating exactly how the human brain processes visual information. The judges awarded him second place in the entire national competition. The prize package included a trip across the Atlantic Ocean. He traveled to Stockholm to attend the 1990 Nobel Prize ceremony. Surrounded by the brightest minds on Earth, the teenager boldly approached organometallic chemistry Nobel laureate E.J. Corey. The high school student asked for a position in the laboratory of the newly crowned laureate. Corey advised the young student to complete the necessary university courses first. The teenager followed the advice. He arrived at Harvard College with the class of 1994. He initially leaned toward physics, but quickly pivoted his attention to chemistry. He studied synthetic organic chemistry under his former idol E.J. Corey, and he studied biochemistry under Stuart Schreiber and Eric Verdeen. He excelled as both a student and a physical experimentalist. He graduated summa cum laude and ranked first overall in his entire class of 1,641 students. He won the de Tour Prize and the Sophia Freund Prize, which represent the highest scientific honors for undergraduates at the university. His senior thesis consumed 119 pages. Working in the Corey laboratory, he detailed the precise physical synthesis of a natural product. He earned the coveted Thomas T. Hoops Prize for this document. Corey later recalled that the undergraduate thesis was absolutely word-perfect and entirely equivalent to a doctoral thesis. The young student approached chemistry like an engineer solving physical puzzles. He utilized a hands-on problem-solving approach that became his trademark. He even applied this analytical mathematical thinking to a university course he taught in Courier House on the mathematics of blackjack. He showed his fellow students how game theory and statistics apply directly to casino probability. He taught them that you win money by betting more when you are more likely to win. He proved to be a dedicated teacher and would later build one of the largest science lecture courses at the university. In 1999, he arrived at the University of California, Berkeley, to earn his doctorate in chemistry. Inspired by collaborators like Gerald Joyce, he shifted toward protein engineering and directed evolution. Gerald Joyce is an American biochemist and molecular biologist known for his pioneering work on in vitro evolution. Lou did not simply want to observe biological processes, he wanted to steer them. His dissertation was titled Expanding the Scope of Protein Mutagenesis. He helped launch the first systematic effort to expand the genetic code inside living cells. His laboratory engineered ways to force non-natural amino acids into proteins. This forced the cells to perform entirely new biochemical functions. Young Lou's analytical mindset famously extended to the blackjack tables of Las Vegas. His skill as a card counter was so advanced that he consistently won substantial sums in his early 20s, utilizing self-programmed strategies. This led to his eventual ban from all Las Vegas tables at the MGM Grand. He later quipped that his only goal was to win enough to buy his wife, Julie, a specific pair of earrings. Lou understood that while one cannot win more hands than they lose, true profit comes from increasing bets when the odds favor the player. He even wrote a blackjack computer simulator while hospitalized. Long before he became famous for rewriting the genome, he spent years inventing powerful biochemical tools. During the early 2000s, his research group developed DNA-templated organic synthesis. He discovered that attaching small organic molecules to complementary DNA strands allows the physical sequence of the DNA to program chemical reactions. He used DNA as a physical scaffold to rapidly generate vast libraries of candidate drugs. Instead of proteins, he used DNA to code for small artificial molecules. This allowed scientists to make lots of combinations of new drugs very quickly. This innovation turned chemical discovery into a programmable high-throughput process guided by genetic rules. Meanwhile, his laboratory expanded the physical hardware of biology. Following his graduate work, he led the major effort to incorporate unnatural amino acids into living cells. Through his rapid evolution platform, he turned viruses into microscopic protein factories. By the middle of the 2010s, his laboratory had introduced dozens of molecular technologies. These included DNA-encoded libraries, new biological selection systems, and synthetic gene circuits. His most significant contribution has been the technique of base editing. Before Lou, scientists knew how to edit DNA. They use a technique called CRISPR. CRISPR has been around for over a decade. The specific technology for using CRISPR for gene editing was developed by Emmanuelle Charpentier and Jennifer Doudna, who were awarded the Nobel Prize in Chemistry in 2020 for the development of a method for genome editing. Standard CRISPR technology operates as a pair of molecular scissors. The system hunts down a specific genetic sequence and completely severs both strands of the DNA double helix. The target cell senses this catastrophic physical damage and panics. It rapidly glues the broken ends back together. This frantic repair process often inserts or deletes random chemical letters at the break site. The resulting scar disables the gene. It stops the gene from functioning, but it cannot precisely correct a spelling error. Liu sought a more elegant solution. Errors in this text can cause cancer and a multitude of diseases. In 2016, he introduced his alternative of base editing. He describes a base editor as a protein that rewrites a single DNA letter at a target site without severing the double helix. He often compares it to a pencil with an eraser. Just as you can erase and replace one letter in a written sentence, a base editor can change one DNA letter to another. A base editor can correct a single point mutation that causes a medical condition. This has shown rapid medical results. For instance, in 2022, doctors at Great Ormond Street Hospital in London employed a base-edited cell therapy to treat 13-year-old Alyssa, who was suffering from fatal leukemia. Her doctors made 3 crucial single-letter edits to donor immune cells. This intervention was a success, with her cancer going into complete remission. Clinicians have used base editors in early trials for sickle cell disease, certain anemias, and other conditions. The vast majority of known pathogenic mutations can now be corrected using these methods. Base editors function like specialized word processors, carefully correcting one letter at a time. They offer cures where none previously existed. Older tools could only disable genes. In 2019, his laboratory introduced another technique called prime editing. Prime editing operates exactly like a search and replace function on a computer word processor. It can remove, insert, or swap whole segments of genetic code with absolute precision. He designed prime editors to take a specific sequence of DNA and replace the entire sequence with a different one. It corrects short misspellings or entire words in the genetic manuscript. This includes fixing insertions or deletions that cause disease. Like base editing, this process operates without making double-strand breaks. It reduces unwanted errors. He explains that you can search for a specific sequence and replace it entirely with another sequence that you want. This offers enormous flexibility for treating patients.
And tonight, I get to join you all to celebrate the man whose work saved my life. So thank you, David.
Liu took his lab breakthroughs and turned them into multiple biotech companies. Back in 2013, he co-founded Editas Medicine. That company focuses on using CRISPR-based molecular scissors to treat diseases that affect the eyes and blood. Later, he helped launch Beam Therapeutics. This group uses its base editors to fight genetic blood disorders. Their clinical trials are curing sickle cell disease and beta thalassemia by essentially rewriting single base mutations right inside patients' blood stem cells. Then there's Prime Medicine, which is focused on his search and replace technology. They're aiming to fix genetic errors that are completely out of reach for simple, single-letter corrections. Then there is Pairwise Plants, which is a joint venture with Flagship Pioneering that takes these molecular tools and applies them to agriculture. They engineer crops capable of resisting extreme weather and fruits that do not turn brown. He is also the brain behind Chroma Medicine, which was formerly known as N-Chroma, and operates on a different level of biology. They develop epigenetic therapies. These treatments turn genes on or off without altering the underlying sequence of the DNA. They target neurological disorders and rare developmental conditions. Another one, called exotherapeutics, focuses on small molecule drugs inspired by epigenetic editing. They aim to enhance genome repair and treat age-related diseases. He is driving at least 23 clinical trials worldwide based on his technology. 15 of those clinical trials are testing base or prime editing therapies specifically. His massive body of work includes over 275 research papers and more than 110 patents. He has served on high-level advisory panels, such as Jason, advising the United States government. His work has earned him the highest honors in the field. Alongside the $3 million Breakthrough Prize in 2025, he received the King Faisal Prize in Medicine in 2022. This is often seen as a precursor to the Nobel Prize. He received the Harvey Prize for Chemistry and Medicine in 2025, which is also called a predictor for the Nobel. He received the Franklin Institute Bauer Award and the 2024 Gabay Prize for Biotechnology. He was elected to the United States National Academies of Sciences and Medicine. He also won top awards from the American Chemical Society and various genetics societies. At the Breakthrough Prize Gala, he was introduced by actress Jodie Foster and honored in person by Alyssa Tapley, the teenager whose life was saved by his technology. He accepted the award while saying that the real heroes are his students and postdoctoral researchers. Starting around 2020, he quietly began giving away his entire Harvard salary after taxes to his laboratory members and students. During the global COVID pandemic, he learned his graduate students could not afford bicycles. He initially tried to help by sending Amazon gift cards so they could buy bikes. The students were wary and thought the emails were scams. He switched to writing personal checks. Now, every Thanksgiving, he donates what would have been his paycheck of over $250,000 to his trainees. His startups have helped him become a multimillionaire entrepreneur who can make ends meet without his academic salary. He even set up a charitable foundation to advance scientific research and plans to fund it with his Breakthrough Prize winnings. He believes scientists have a responsibility to society. He emphasizes the importance of rigorous safety and ethics, noting that regulators do not kill anyone but ensure new therapies are safe. Outside the lab, his hobbies remain eclectic and playful. He built a 6-gram carbon fiber model airplane called the Wisp. It flew at a leisurely 1 mile per hour, solving an engineering puzzle about slow flight. He built a homemade gadget dubbed the Mousepult using Lego bricks and a burglar alarm heat sensor to detect cats and launch tiny toy mice. He has 2 pet Bierman cats. During his undergraduate years, he hung out with the Naughty Dog video game team, who later became famous for Crash Bandicoot. Before his scientific fame, Lou was a prolific professional Street Fighter II player. He was described by Naughty Dog co-founder Andy Gavin as a savant who was insanely good at the game. He served as the lead tester for Naughty Dog's 1994 fighting game, Way of the Warrior. Additionally, he provided the voice for the character Qin the Dragon Liu in the same title. He is a self-described introvert who enjoys puzzles and tinkering. His office contains a life-sized Iron Man figurine and shelves of gemstones and inspirational science photos. He is married to Julie and has 2 daughters, but keeps a low personal profile. Today, he continues pushing gene editing into new territory. At the 2025 Wired Health Summit, he announced his lab is advancing a disease-agnostic gene editing strategy. They are developing one editing approach to treat many different conditions, regardless of the specific mutation or tissue involved. They are building smaller and more efficient editors. They are optimizing delivery into human organs and exploring long DNA insertions. They want genome editing to be as easy as downloading an app to fix a genetic bug. Prime Medicine has advanced a prime editing therapy for sickle cell disease into trials. Beam has shown that base editing can boost normal hemoglobin. Despite all he has achieved, he is in no mood to slow down.